Hello and a warm welcome to everybody joining us for today's last session of the opening day of 2021's D3, Drug Discovery, Development and Diagnostics. It's our annual event. Thank you very much to all the attendees that have stayed with us since this morning. But that brings us to the last session for the day, which is going to be our second disease focus of the day, which is on Leishmaniasis. I'd like to introduce the first speaker to the floor, which is Professor Maria Paola Costi from the Drug Discovery and Biotechnology Lab at the University of Modena and Reggio Emilia Unimore in Italy. Thank you very, very much, and I look forward to over to you, Maria Paola.
Okay, thank you, Cameron. Thank you, and thank you to Marianne and the organization to allow me to present today our work and general introduction that is here. I hope you can see my slide. Okay, WHO roadmap for the next 10 years was announced recently, and they were looking back at the 9 previous years of work they have done, and it was very relevant to see, to find out 2 important facts. One relevant fact is that the ISNTD interventions that was done was estimated in terms of benefit for individual affected individual in terms of let's say dollars, $25 per dollar invested in preventive chemotherapy. These are numbers, but we should translate this in terms of quality of life and benefit for the patient. So, which kind of intervention they have done? It was done on health management and organization in the countries considered, and also in terms of therapy management. So, if this reorganization and care about the general health, let's say management and organization, do we need new drugs, or the drugs we have nowadays are let's say enough, and we can implement somehow what we already have? The answer is that we need to provide innovation in this field, because and to consider that this is the Leishmaniasis is an infection that deserves to be considered as all the other neglected tropical disease, and deserve innovation, and we have to do that. We have to bring innovation in this area. And so, but thinking in specifically about Leishmaniasis, we know that this is not a unique disease, but we have different manifestations of the same disease, and we have cutaneous and visceral and mucocutaneous. But also we know we are in presence of people infected but asymptomatic. So they do not show symptoms, and they do not show they are not sick in some way, but they bring and have the parasite basically. And so another consideration is that we have this aspect related to reservoir of infection. In this case, this is a vector-borne disease, and dogs, so domestic animals, but in particular dogs, are a reservoir of the infection. They are affected by canine leishmaniasis. And this aspect is relevant. In fact, we consider, if in the sense that from the therapeutic intervention point of view, we need to keep in mind this, let's say, continuous presence of some reservoir in the close to humans. And in fact, and the other aspect is that if we look at the world distribution map, in particular, let's say one of these visceral leishmaniasis, we see that this infection is distributed in the southern part of the world, but climate changes is, let's say, changing also the general distribution, and so and the epidemiology. So what I would like to point out is that we have, of course, a huge number of countries affected, 98 countries. This is a number, and 2 million cases per year. But and these numbers today are more relevant to us because we have the experience of COVID-19. So we understand what it means, 2 million cases of infection, of people infected. But part of that, look at the coincidence map. What is it? This is a map in which we overlap the distribution. This is the case of Mediterranean countries. And so we have a superimposition of the human, let's say, infection superimposed with the canine leishmaniasis that are represented by the bars. So this tells us that we are in presence of a One Health problem. So when we cure and we want to cure human leishmaniasis, we also need to keep in mind that we have a reservoir, we have dogs, and so we need also to cure this. If we want to do to make a full, coverage of and the elimination of the infection, we need also to consider this aspect. So it is a typical One Health problem, and in principle, it would require One Health approach. Going to the focusing on on drugs and therapy, there are there are important aspects to consider. The speaker after me will talk also more deeper inside. We'll go deeper inside a few aspects, in particular clinical aspects. But what I would like to say that we have a low number of drugs, and these drugs are not, let's say, have problems because they develop resistance. Some of them are toxic. Some of them, like mutaflozin, for example, they come from a repurposing approach, and because it was used as anti-cancer. And here it is. But it is the only oral drug that we have nowadays, and it's relevant to consider this in terms of a general repurposing approach that all people would like to take. But how to respond to the quest for new leads and drugs? So we need to consider that we have new targets. Innovation is brought through new targets consideration, and we have new targets available, new technologies. Some of them are very costly and so expensive, and we can ask ourselves, how can we approach these kind of new technologies like omics technologies, artificial intelligence, for example. It's very, up to date nowadays, but it's also really very important. And high throughput technologies. There are, considering that we are in a global research network, nowadays it is possible to be in this situation. So we can also consider access to important centers that are available for at a low cost or no cost for interesting projects. So we need to also, for example, in Europe we have some of them, but these are available also in other parts of the world. What I would like to show today is an example of one project that we have developed in our laboratory network of labs. in particular from the one previous project, and we have now a follow-up of that, and in collaboration with a US company working on artificial intelligence. And what we have done is was to consider a database of approximately 500 compounds, and these compounds were tested against 6 parasites, including Leishmania parasites, and panosomes, and Mycobacterium tuberculosis. So we did a broad spectrum testing, and also we evaluated the let's say the armatox properties, cell-based. And all these numbers, let's say, and properties are were determined experimentally, but and at the same time, in parallel, we did evaluation, clusterization, and organization of the project in terms of data analysis. And here is the workflow. I'm not going deeper inside, but please see the starting point. It is a database. And we did screening, we performed screening and then data analysis up to the identification of an in vivo compound that was valuable for in vivo studies. So in particular, the library was first clusterized, first devised, so analyzed in terms of chemical structures and scaffolds. And we finally were able to clusterize based on molecular properties. This helped us to recognize scaffolds that is of course very important in terms of next step evaluation, results evaluation. Here is the heat map reporting on the left, where all this red is, you can see all this red spot is, and in terms of, on the right, we have all the ADME properties that we have evaluated, and we were able to select some compounds belonging to specific scaffolds that are reported here. And from here, we develop a new library through medicinal chemistry classical approach, and we perform the same analysis in order to validate our findings. And in fact, we tested again and confirmed that the new compound were less toxic and more active. And we were able to identify one compound in particular and some derivatives, but this is the best one. And this was, let's say, was tested in vivo. This was, all this work was performed used on the basis of this, let's say, artificial intelligence method. It was a machine learning method that were utilized, combined with isotope screening, because all the screening was performed with the HTS platforms. In particular, we use the screening port at Fraunhofer in Germany, in Hamburg. So this was the final result of our work, but this is just an example of what it can be done using these kind of technologies. Another important technology that can be we have used recently is proteomics, mass spectrometry proteomics, and this is very helpful. This is just an example, but of the final, let's say, network that we could achieve. But in the case of miltefosine treatment of Leishmania cell, promastigote in this case, and but what can omics be helpful to do? Which kind of advancement can bring? We know that we, of course, have information about new targets, about the mechanism of our new compounds or drugs, and also this opens the way to the identification of biomarkers. And in this way, we can enter into the field of diagnostics, right? So Okay, just to focus also to drug repurposing. This is another solution for facing drug discovery problems and bringing innovation. Looks like not so much innovative to start from in the field of drug repurposing, but we need to consider that despite the concept, the basic concept is to start from, let's say, a known drug and to try to recycle this new drug in a different field, in a different therapeutic field. This requires some work. So we are in this, for example, in this picture, you see the full discovery process. And okay, in principle, we think that we can start very, very advanced. We are very, very advanced with respect to a hit and lead search, but we need to be very careful. And we have a demonstration recently with the COVID-19. We did this. We try to repurpose new drug, but need to be careful because the drug profile has to be reviewed carefully, and maybe it is necessary to modify something. Of course, it is faster in principle than starting from hit and lead optimization. But still some work has to be done. So this is some considerations that come from recent experience. Now, in principle, I could talk about clinical landscape, but I know that we had the next speaker will talk specifically about the clinical landscape. So I will skip my slide about this. Coming to the final slide, in which I can focus on some actions that are ongoing at the European level, and we have many of them. And now we have the starting of the new Horizon Europe, so there will be more, we hope. And this is to thank all the collaborators belonging to this group, and also to new collaboration we have started from that. And this to remember that we have a lot of work to do, too many roles not taken yet. So thank you for your attention.
Thank you very much, Maria Paola, for that fascinating landscape. It's thrown up a lot of questions, which we'll come to. We're definitely going to have the Q&A at the end of this session, because it's the final session. We can absorb the time. We're getting a lot of questions, a lot of varied country spread in terms of the inputs. There's questions. So from Tunisia, we have from India, Brazil, Nigeria, the UK here, various countries asking questions in terms of that particular presentation. So thank you very, very much for that. I thought it was fascinating, the use of this omics and AI as well, new horizons in terms of the effort there. Some questions coming in on about in terms of the various pathways. Someone there is involved in purine salvage pathways, asking quite a few questions, and we will definitely come to those at the end. Thank you very much for that. I mean, that brings us to our second. We've heard the molecular diagnostics kind of, the discovery all the way to clinical landscape by Professor Maria Paola Costi from Unimore. We thought it would be a great place now to bring in the head of visceral leishmaniasis at the clinical program, again at the DNDi, the Drugs for Neglected Diseases Initiative, Dr. Fabiana Alves, who heads up that clinical program for visceral leishmaniasis, to talk more about the current and future perspectives. We heard a little bit about the future perspectives for the development of new treatments for visceral leishmaniasis.
Thank you so much. So first of all, thank you very much for the invitation to be here today, participating in this ISNTD seminar. It's really a pleasure to be here with all of you. So I was asked to present on the DNDi strategy for the development of new treatments for visceral leishmaniasis, and I'm going to give you the view of current and future perspectives for the drug development. So Maria Paula helped me a lot because she gave a lot of this initial introduction to the disease. Here, I think my main message from this first slide is to remind you that among the different forms of leishmaniasis, the most severe form is visceral leishmaniasis, which is a disease that is fatal if not treated. And here you have on the right the picture of this child with VL. So usually they come with very enlarged spleen, liver. Usually they are very malnourished, pancytopenic. So it's really a disease that you have to act fast and treat these patients as soon as possible. The other important point I would like to make is that half of these patients affected by VL are children. So going now to the distribution of the disease, and also you saw already this map. This is the WHO map for the distribution of VL in the world. There are really three main endemic regions that we have. So South Asia, East Africa, and Latin America. So historically, South Asia was really the region where they had large majority of cases, in the world. But after there was political decision to implement the elimination program, now numbers are decreasing steadily year after year. So they are really moving towards elimination as a public health problem. And I think this is really a very good example that you have to look at and see if this could be also implemented in the other regions. East Africa is currently the highest burden region in the world. As you can see, countries affected are mainly in the Eastern African region. So it's Sudan, South Sudan, Ethiopia, Kenya, Uganda, and Somalia. And even though, in these two main endemic regions, the disease is caused by Leishmania donovani, it is an anthroponotic parasite. But even though it's the same Leishmania donovani, as you can see from the boxes, when you look at the first line treatment and second line treatment, they differ between the regions. And we think that this could be related to the parasite factors and host-related factors. But the truth is that you don't have one treatment that is successful for all of the VL patients. So the message is that you always have to adapt your treatment to the reality in each of the regions. And in general, it is more difficult to treat a VL patient in Africa as compared to Asia. Now, going to the other region that also we have a lot of cases, it's in Latin America, where more than 95% of the cases occur in Brazil. As mentioned by Maria Paula here, the disease is caused by Leishmania infantum. It is the same parasite that causes leishmaniasis in the Mediterranean region. It is a zoonosis there. Dogs are important reservoirs, and indeed this brings a lot of challenges to the control of the disease. In Brazil, currently the first line treatment is antimonial, but they are in process of changing to the Ambisome treatment. So these are the drugs that we have today to treat leishmaniasis. You probably are familiar with all of these drugs. We have the old antimonials that still have been used, except in Asia where there is resistance. Then we have liposomal amphotericin B, paramomycin, and mutafosine. And as mentioned before, all of these drugs, they have limitations, mainly associated with toxicities associated with these treatments. All of them, except mutafosine, are parenteral drugs that require IM or IV infusion. And in the case of amphotericin B, we need cold chain, which makes life a little bit complicated in the field, requiring really logistics to be able to implement this treatment. And if you look at the only one which is oral, mutafosine, it is certainly an improvement because it is oral, but we have, the issues of poor tolerability. A lot of patients have a lot of vomiting when they are treated with mutafosine. And I think the most important is the teratogenicity associated with mutafosine, which makes it very difficult to implement for women of childbearing potential in the endemic regions. So this is the slide where we talk about the NDI strategy and how we see the development of new treatments. So for each and every disease that the NDI is working with, the first thing we ask when we have to implement a new strategy is really what are the patient's needs. So it's through consultation with the main stakeholders and developing what we call the target product profile that we are going to then identify which is the treatment that is needed for this patient population. So as described in the WHO NTD roadmap, one of the critical actions clearly is that we need to develop more user-friendly treatments. Examples are oral treatments that would be able to be implemented in primary healthcare settings. So the DNDi strategy aims to deliver safe, effective, short course oral treatment for visceral leishmaniasis. So VL is the main patient population. However, we have to look at other types of manifestations like post-Kala-Azar dermal leishmaniasis. I'm going to talk a little bit about this later on. And also those who are very difficult to treat, the special population of co-infection patients who have both HIV and VL. And through the programs, we are always building and maintaining lots of partnerships in the endemic countries. So we build platform and consortiums that facilitates the clinical activities in the field, but then later on also aim to ensure access to these new tools. In this graph that you see here in the bottom, this is the DNDi portfolio for leishmaniasis. Each of these boxes represents one project. So from the beginning, we had short and long-term objectives in our strategy. The short-term strategy was to look at, okay, these are the drugs we have to treat leishmaniasis today. They all have limitations. Is there a way to improve on these treatments? So most of the new treatments that were developed were focusing on combination treatments. So by combining, you can reduce the treatment duration, usually improve safety and maintaining good efficacy rates for these drugs. So we have developed with partners the current first-line treatment in Africa, SSGPM. There is also the combination treatments in South Asia. Paramomycin and miltefosine is indicated as second-line treatment. We also participate in the effectiveness study for rollout of single-dose amisome. So these are achievements. But this is not sufficient, right? So right now, I'm going to give you more details about studies we are performing currently in Africa for development of paramomycin miltefosine for VL, the study we did with partners for HIV VL, and also studies on PKDL. But I'm going to give you more details in a while. And then, so these are the, treatments that we expect to have results this year, to be implemented maybe in the coming years. Of course, everything depends on the outcomes. We are positive that we're going to have good outcomes, but we have to wait and see. But really, the change in how we treat visceral leishmaniasis, we expect to come from the left side of the portfolio. Each of these boxes represent one new chemical entity. And you heard a lot from Maria Paula, the work it takes. So over the past 10 years, not just DNDi and many other partners invested a lot on the identification of new compounds. And fortunately, today, we have a good portfolio of different classes of compounds with different mechanisms of action that we believe will be able to deliver the oral, safe, effective treatment that we need to be implemented at the PHC level. So now I'm going to take you through a few details of the projects that we are conducting. So just here, as I said, Africa, we have a lot of attention to Africa because it's the highest burden region. And the previous treatment there was SSG alone for 30 days. So there is an improvement with SSGPM with 91% efficacy. But these still have 17 days of 2 injections. You can see here the picture. SSG is quite a high volume. Paramomycin injection is quite painful, so it is a burden for the patient, right? There is also the toxicity associated with SSG that remains there. Patients that are above 50 years old do not tolerate, cannot take this treatment. The same thing for HIV-VL. So looking at alternatives, the one that looked more promising was to replace toxic SSG with oral mutafosine. So the NDI did some studies, and first thing we observed was that when we use the mutafosine conventional dose that is highly efficacious in Asia, in Africa, we didn't have the same level of efficacy. We had only 72% efficacy, and especially children less than 12 years old, they had lower exposure of mutafosine and only 59% efficacy. So then there was a lot of work that was done by our partners suggesting that we had to actually modify the regimen for mutafosine in children, and then by using an allometric dosing, we were able to achieve 90% cure rate in children, and this is now the strategy that we are using for the new study. So paramomycin mutafosine is a treatment that is highly efficacious in Asia. With 10 days, you can get more than 95% efficacy. And it looked quite attractive in Africa, especially for the use in children. So we have launched a study back in 2018. It's a large phase 3 trial that involves partners in Ethiopia, Sudan, Kenya, and Uganda, where we are assessing this combination. But here we had to adapt, as I said, for Africa, longer duration, 14 days, and allometric dosing for mutafosine, aiming to achieve more than 90% efficacy. And this treatment now we're going to be comparing with the standard of care SSGPM. Fortunately, despite all the challenges of COVID, we were able to complete recruitment in May, finish the follow-up. We need to do follow-up at least for 6 months for VL. This was completed in December. Now we are working hard on the data, and we hope that by beginning of Q3, around July, we should have the results of this trial, and hopefully with good results, as I said, mainly for children would be a very good alternative. Now, I was also asked by Cameron to talk a little bit about HIV-VL. So when we have patients that have visceral leishmaniasis, but at the same time are co-infected with HIV, these are the most difficult patients to treat. These are really patients that require very special management. They have usually poor treatment outcome, higher risk of mortality. And once you treat them, they have to be followed up because they have high risk of relapse, especially when they have low CD4 count. So it is very important that this patient has a holistic approach. You treat not just the VL, but also ART that is effective for the patient, and also treating other comorbidities that are very common in this patient population. HIV-VL is reported in 35 countries. Some countries with highest rates are Ethiopia, where we have 20% of the patients with HIV-VL co-infection, Brazil, and India. And to bring here the data, so based on the experience of MSF working on the day-to-day, having to deal with these patients that were very difficult to treat, they tried everything they could, and then there were very good, promising results with the combination of mutafosine. So the NDI with MSF, University of Gondar, Addis Ababa University, and ITM, we planned a randomized controlled trial in Ethiopia to assess the current treatment recommended by WHO, Ambisome 40 milligram per kilo, but also the combination Ambisome mutafosine. So to summarize, we had actually very good results with this study. We were able to achieve 91% efficacy at the end of treatment. And as I said, combined with ART, secondary prophylaxis during the follow-up, especially for those who have low CD4 count, and managing other comorbidities, that's how we think we would be able to improve the treatment for this patient population. So all this data was revised by WHO in the guideline development group back in October last year. And now we are expecting that the new guidelines on HIV-VL are going to be released around Q2, both by WHO, but also by the government in Ethiopia. And there is also the similar study which was done by MSF in India, having similar results. So this is the summary for HIV-VL. Now, this is one slide where I try to summarize post-Kala-Azar dermaleishmaniasis. This is a condition that happens after patient is treated for VL. So depending on the region, like in Asia, 5 to 10% of the patients are going to develop PKDL, usually average 2 years after treatment. In East Africa, the country with highest rates is Sudan, where estimations is up to 50% of the patients can develop PKDL after being successfully treated for VL. The manifestations can differ. Also in India, usually they have macular presentation, as you see here on the left. In Sudan is the central picture with papular manifestation, but you also can have nodular manifestation of PKDL. So these are patients that are usually healthy individuals other than the skin rash. The problem is that they don't seek for diagnosis and treatment. They stay with this rash for a while, and now we know that they are important reservoirs of the disease. So there is a lot of attention for the need to identify and treat PKDL. However, the treatment we have today are really not appropriate for this patient population. So as you can see, antimonials up to 2 to 3 months with high risk of cardiotoxicity for a disease that is not life-threatening. Mutafosine for 12 weeks in South Asia, very difficult to implement, especially for women of childbearing age. And also when in Sudan they use Ambisome, they have to use very high doses. So clearly there is a very important need to improve treatments of PKDL. And some years ago, we started 2 studies, one in Asia, India and Bangladesh, and another one in Sudan. And again, we were able to complete recruitment. Now we are in the follow-up phase. Results expected this year. Again, we hope that at least this could be an improvement. These are combination treatments which are going to be shorter and hopefully can have an alternative in the short term for PKDL. So here, this is the summary of studies that are part of the short term objectives and strategy, which we could deliver new treatments very soon. Results are going to be available this year, so hopefully with good results. But then now moving to what is going to be really the future of treatment for visceral leishmaniasis. And as defined by the WHO NTD roadmap, really the way we see the future of VL treatment is to focus on the development of oral, safe, effective treatments. So moving from injectable toxic drugs with variable efficacy requiring hospitalization to treatments that are oral, well tolerated, and therefore could be integrated at the PHC level. So again, I bring here, these are the portfolio with the different compounds. There was a lot of work done to be able to have this portfolio by the NDI and partners. Today, there are 5. In fact, there is one more, 245, done by GSK, has moved to phase 1. So we have 5 compounds that are currently being tested in healthy volunteers. The one that is more advanced is the one that LX0408 identified by Novartis. We have a collaboration with them, and we hope this year we are going to submit the first phase 2 trial to start study in patients. So there is a lot of expectation that with this very rich portfolio, we will be able to deliver pan-geographic oral treatments that will address the issues of safety and efficacy in all of the different regions. We believe that, as I said, the treatment will have to be tailored to the needs and characteristics of the population in the different regions. So maybe for some regions we could say that maybe monotherapy would be sufficient, but maybe for others, combination would be required, and that's why it is important to have many compounds that we'll be able to test, right? Byron is going to tell you more about immunomodulators, especially for complicated forms of CL. And then the other aspect is that we are putting a lot of attention in children. As I said, 50% of VL cases are children, so we are working on a pediatric plan to include children as early as possible and have the treatment adapted to this patient population, as well as women of childbearing age. So by having oral drugs that are safe and also associated with diagnostics that can be deployed at PHC level, we think that this is the way that we'll be able to integrate VL treatment with other diseases at the PHC level. So really changing the way we currently treat these patients. Another aspect that there is a lot of attention in the NTD roadmap is about prompt access to diagnosis and treatment. So by having a treatment that you can integrate at PHC, you would be able to reduce the time between onset of symptoms, access to treatment, therefore reducing morbidity, reducing mortality, which is the main indicator, and also breaking the cycle of transmission. So this is what we think will be, the future for VL treatment. This is it. Thank you so much. I hope I didn't take too long, Cameron.
Thank you very, very much, Fabiana. I'm very happy that you took a long time, because I was delighted to see you included the HIV co-infection angle in there as well. We think it's very interesting that you did that, simply because the WHO roadmap is now talking about reaching out across different disease states. And most of the funding in global health goes to HIV kind of crowd. So I'm wondering how that's going to impact on that. I'm sure we'll come to that in the questions in the Q&A, but we're getting some really good, strong responses in the chat boxes. An excellent presentation, and it was excellent. A very interesting point you made about this, the PKDL reservoir, because somebody else was asking earlier, where are the disease reservoirs? So an increased kind of surveillance net, will this be part of all of your rollout? What I took from that was so much thought goes into a DNDi, program, whether it's regional thought or the partnerships on the ground. You really do put access there, right there, and help redefine that. So thank you very, very much for that indeed. It's wonderful.
Thank you very much.
And I think that brings us on, no problem. I think that brings us on very nicely to actually your colleague. So Dr. Byron Arana, we've heard of the visceral Leishmaniasis clinical programs and that kind of landscape around that from Professor Maria Paula Costi and Dr. Fabiana Alves of Unimore and DNDi respectively. We wanted to kind of look at the cutaneous Leishmaniasis scenario, and so we're very honored to be joined by Dr. Byron Arana, the head of the cutaneous Leishmaniasis program at the DNDi, from bench to cure.
Well, thank you very much for the invitation. For the audience, they are going to hear some repetitions, because obviously the two previous speakers have included some of this information, but I believe it doesn't hurt to repeat some of this data. So typically, give me a sec. Yeah. Typically, when talking about cutaneous Leishmaniasis, we refer to cutaneous Leishmaniasis in the New World and Old World. This classification is based, besides the geographical distribution, of course, in the species of Leishmania causing the disease, which are different in both parts of the world. Each of these parasites have a particular life cycle involving different vectors, reservoirs, and clinical manifestations. Some of these infections evolve to a more complicated clinical form, such as the Leishmania recidivans, which is usually associated to infections to Leishmania tropica, or mucocutaneous Leishmaniasis, which is associated with Leishmania vianea parasites in the New World. It has been estimated that every year there are more or less between 0.7 to 1.2 million of new cases of cutaneous Leishmaniasis only, which is equivalent to more or less one case every 30 seconds. By 2016, WHO reported that 87 countries were endemic for CL. And based on the number of cases reported in 2018 to WHO, it was found that 12 countries contribute to approximately 90% of all cases globally. It worth mentioning, however, that only around 70% of countries considered endemic for CL report routinely to WHO, and some of the countries who have very high numbers are not routinely reporting to WHO, sadly. No prophylactic vaccines against CL are available, and the drugs we are currently using have serious limitations, making CL altogether one of the most neglected diseases among the NTDs included in the WHO list. As you can see in this graph, the number of CL cases reported to WHO during the last 20 years has increased by 5-fold, more or less. From 50,000 cases in 1998 to 250,000 in 2018. Even though these numbers are partially because more countries are now reporting to WHO, the major contributors to these numbers include the emergence of new CL foci, outbreaks which are reported constantly every year, the high number of cases reported in refugees, camps, and displaced populations. The Syria example is one of the best examples, the problem in Syria, and an increased number of CL cases reported among international travelers. This slide summarizes the current treatment recommendation for CL. Basically, treatment is recommended based on the clinical presentation of the disease and the species of Leishmania causing the infection. Treatment recommendation goes from only observation, as in the case of lesions, small lesions caused by Leishmania major or Leishmania mexicana, which are considered to be self-cure at least around 50% of the cases in around 12 weeks, followed by topical treatment for those patients with small lesions in size and number, usually no more than 4 lesions of no more than 4 centimeters in diameter. Systemic treatments are recommended for patients with large lesions in size and number, or lesions in face or anatomical areas where a topical treatment is not feasible. And finally, there are several combinations recommended, which are reserved for special cases of patients with the Leishmania recidivans, for example, or those patients who don't respond to monotherapy. Regarding topical treatments, the ones recommended by WHO includes liquid nitrogen or intralesional antimonials, either alone or in combination. These treatments are applied to 1 or twice per week for several weeks, sometimes up to 15 weeks. Thermotherapy, even though it's much simpler, requiring only 1 to 3 applications, is sadly not widely used for different reasons. The efficacy of any of these treatments is variable, and in most cases around 70% only. A cream based on paramomycin has been for many years mentioned in WHO treatment guidelines and in other treatment guidelines. However, such cream is not registered and not commercially available yet. So no patients are being treated with this cream. That's why I cross it out with this red line. Regarding systemic treatments, Fabiana has already mentioned the advantages and disadvantages mainly disadvantages for the drugs we are currently using. We continue using drugs that were developed for visceral leishmaniasis and for other conditions. It's worth mentioning that no drugs have been specifically developed for cutaneous leishmaniasis. What we are using is because we have inherited from other diseases. Antimonials, either meglumine antimoniate or sodium stibogluconate, are the more widely used. Treatment consists of 20 days of intramuscular or intravenous injections. More of the patients, around 60%, develop electrocardiographic abnormalities or liver or renal enzymes. And its efficacy varies from 50 to 90%, depending on the species and the clinical manifestation. Miltefosine, as mentioned by Fabiana, is so far the only oral drug available. It needs to be taken for 28 days. Nearly 60% of patients experience nausea and vomiting. And it's, as it was mentioned, the main problem is the teratogenicity, which makes, if you are treating women in childbearing age, they have to use contraception up to 3 months after the end of the treatment because the long effect of miltefosine. Pentamidine is recommended only for Leishmania guianensis infections, while the use of amphotericin B the experience is quite limited given the cost and is mainly used as a rescue treatment in some cases. So as you can see, there is a big gap here in terms of the need for a short, safe, and more field-adapted treatment for cutaneous leishmaniasis. And this applies not only for cutaneous leishmaniasis, but for visceral leishmaniasis, as previously mentioned by Fabiana as well. So in response to these needs, PNDI, in consultation with experts on CL, researchers, and other key stakeholders, developed a strategy aiming to cover all possible scenarios given the current treatment recommendations. The strategy aims, as similarly to BL, to develop a short, efficacious, safe, and easy-to-use treatment for all forms of cutaneous leishmaniasis. And as in the case of visceral leishmaniasis as well, we have split the strategy in the long term and the short term. In the long term, the NDI is looking for opportunities to develop a topical treatment for those patients with uncomplicated CL, oral treatment for patients with large number of lesions or those who do not respond to local treatments, and an immunomodulator for cases where the lack of proper immune response by the host has been implicated in the severity, the chronicity, or the treatment outcome of the disease. We acknowledge, however, and that was mentioned by Maria Paola at the beginning, that developing of this treatment from the screening to registration takes a long period of time. Therefore, in the short period of time, it is proposed to see if there is an opportunity to better use any of the current approved treatment options, but in combination. Following different approaches, but always avoiding duplication or efforts and prioritizing the collaboration with other groups, during the last years, the NDI has been able to advance in its ECL program. In relation to the improving existing tools, the NDI is testing the combination of a single application of thermotherapy, 50 Celsius degrees for 30 seconds, plus 3 weeks of miltefosine. We completed a phase 2 study showing the superiority of the combined approach when compared to thermotherapy alone. Now we are embarked in a phase 3 study in 4 countries in Latin America, aiming to demonstrate the non-inferiority of the combination of the thermotherapy plus miltefosine when compared to the standard monotherapy treatment, either antimonials or miltefosine. Results of this study are expected by mid-year next year, by Q2 2022. Regarding the development of topical treatments, we know that there are several groups working on this, on different formulations. So rather than duplicate efforts, it was decided to better enter in communication with these groups and see if there is any opportunity to support and collaborate with them. One of these groups, by the way, is the one leading by Professor Dolores Carrer, who will be the next speaker in this symposium. Concerning oral treatments, Fabiana has also already mentioned the different new chemical entities that are being developed by the NDI and other groups. What I'm going to mention here is that these NCEs have shown to be very active against Leishmania strains causing visceral leishmaniasis, but also cutaneous leishmaniasis. So our plans are that as soon as the phase 1 studies are completed, which are similar for both conditions, we plan to initiate phase 2 studies testing these compounds on CL patients. As regard to the immunomodulators, the NDI is developing a TLR9 agonist. It belongs to a class B CpG and is named D35. D35 is a synthetic DNA molecule developed in the FDA laboratories in collaboration with Osaka University. It stimulates mainly the plasmacytoid dendritic cells, producing IFN gamma through the activation of the natural killer cells, monocytes, and dendritic cells, and secreting IFN alpha and beta, but having no effect on B cells. CpG D35 is proposed to be used in combination with an antiparasitic drugs, either the existing ones or the ones that the NDI is developing together with other partners. We are planning now, we were able to complete all the preclinical and the CMC studies of this D35 last year, and now we are organizing the phase first in human study, the single ascending dose study. We are hoping to initiate in Q2 2021. Now I'm just going to a few words regarding the recently launched 2030 WHO NTD goals. As you for sure know, CL is targeted for control only. The goal by 2030 is that 80% of all cases are detected and reported, and 95% of those cases treated. Using the numbers of new CL cases per year estimated by WHO in 2010, it means that by 2030, we will need to be treating between 0.6 to 1 million of new cases of cutaneous leishmaniasis every year. And based on what we know right now, the only way of achieving such an ambitious goal is by having in place several tools, mainly a safe, efficacious, and short and easy to use treatment for cutaneous leishmaniasis. There are, of course, other needs which I'm not going to discuss in details in this opportunity, but I believe it's worth mentioning them, including the need of having a point-of-care diagnostics, which I understand tomorrow is going to, there are going to be several talks on diagnostics, development of better system for identification and treatment of patients, and of course, better reporting systems. Several studies have shown that the number of cases reported by the national programs are much lower than the real numbers. So system for early identification, treating and reporting CL cases are needed to need to be strengthened if the countries want to achieve the 2030 goals. Finally, a few words about the other topics which are of paramount importance, but still not receiving enough attention and support. First of all, the long-awaited prophylactic vaccines against cutaneous leishmaniasis. Many studies testing different vaccines have been conducted. We are now testing candidates for the so-called third generations vaccines, but still it's not clear if any of these vaccines will complete the cycle and being registered. The issues of mental problems associated with the skin NTDs and its inclusion in the DALYs calculation has recently been to the discussions. However, we do not know at this stage which is the best methodologies to assess this mental problem burden. And of course, we also don't know exactly how to manage this situation. Even when we are developing different treatments for cutaneous leishmaniasis, most clinical studies you need to keep in mind are conducted in adult population. So additional studies, which implies additional time, are needed to determine the safety and efficacy of any of these new treatments in children, pregnant women, patients with other comorbidities. Similarly, there is not much information about how to treat all the outbreaks that are being reported on a regular basis. The issue of access was discussed today in the morning in one of the presentations, and we constantly hear of shortage of anti-leishmania drugs, the use of counterfeit medicines, and other issues hampering the proper and opportune treatment of patients with cutaneous leishmaniasis. Finally, we believe that more R&D on vector and reservoirs are needed to better characterize the life cycle of the parasites and hopefully to develop better preventing and prophylactic treatment interventions. So, with respect to treatment, we strongly believe that a promising and brighter future will come in the following years when the current drugs, which are toxic and requires many weeks of treatment, can be replaced by a safer and more efficacious new drugs. Thank you very much. We would like to mention the support from our different partners, including, of course, the UK, Swiss, and German governments, GHIT, and another supporters among other groups worldwide. Thank you very much.
Thank you very, very much, Byron there. A fantastic overview of the cutaneous leishmaniasis kind of landscape there, and some real strides forward in terms of the DNI's work in topical, oral, and immunomodulatory mechanisms in terms of your movement in that. I thought you had some very interesting gaps that you alluded to in terms of the mental health gaps, the gaps at the vaccine level, towards the end of your presentation. I thought that's something we can pick up in the Q&A, but I thought it was fantastic. We wanted to really show leishmaniasis as a whole. That's why we wanted to include CT as well. So we're very, very thankful for that overview, Byron. It was very, very useful. You mentioned some of the great partnerships that you've got going already. Obviously, GHIT, they seem to be everywhere. We had them on both sessions this morning for that particular reason. The various governments as well. But you also mentioned Professor Dolores Cara's work at CONICET. I think that's a great segue into this particular part of the session. I think without further ado, I'd like to hand over the floor, introduce to you Professor Dolores Cara from the Biophysics and Nanotechnology Lab at INIMEC CONICET, Argentina, joining us here actually to talk really about the topical treatments for cutaneous leishmaniasis. I know that Byron had spoken a bit about the oral treatments as well as the immunomodulatory front that they're opening at the DNDi. But in terms of topical treatments, I'd like to hand over the floor to Professor Dolores Cara.
Thank you so much for the presentation. And to start right away, well, here I put in red my province in Argentina so that you know where I'm based. About treatment for cutaneous leishmaniasis. Current therapy is, as Byron already explained, mainly very toxic, and most of the complications coming from this therapy arise from the toxicity. The most useful, which is oral miltefosine, has this main disadvantage that you cannot use it in children, you cannot use it in women of childbearing age without concurrent things not to get pregnant. We are in dire need of something new, and I really like the idea of topical treatment because, well, it has some obvious advantages. We will talk about the disadvantages later. So mainly the advantages are derived from the low toxicity because the drug would only work at the site mainly. It would also enhance patient compliance. It will be easy to use. It will have decreased side effects, and it will be useful in a context with low medical infrastructure, which is most of the regions where the disease is endemic. And also, since before they were talking about the amount of children that are affected by a visceral leishmaniasis. In cutaneous leishmaniasis, also children can be up to 70% of the patients, depending on the region. So it is really important to be able to have a treatment that is good enough for children and women. So where are we right now? This that I explain now, it's been obvious for many years. There has been a lot of work done on trying to develop a topical treatment for cutaneous leishmaniasis. However, there is only one, and I put here paromomycin, but I hear somebody before said that it is not commercially available. So maybe I should also cross it with red, like Byron said. Paromomycin is not even commercially available, and right now there are, as I know, only 2 topical formulations in clinical trials, one of paromomycin and gentamicin, and then there is a liposomal amphotericin B that has gone through the first phase of a clinical trial. So it's really scarce what the amount of new things that are there, right? There are some new topical formulations with preclinical data available. Most of them have been published in the last couple of years, but of course only on mice, and most of them use miltefosine, which has proven to be very effective, but the literature has a very so each paper has worked with only 1 or 2 variants of leishmania, which is we know that the different species react differently with to the drugs, so it is really important to have these treatments studied against all the possible species of the parasite. So these are 3 very good publications, and here there is one, and all of them have used some kind of penetration enhancer, so either ultra deformable liposomes or DMSO or fluid liposomes. So I want to talk about our work, which is has also been centered on miltefosine. We started with this 4 or 5 years ago, and we published it just now. So here. So what have we working on? We've been putting miltefosine into liposomes, right? We tried different liposomes, and we studied cytotoxicity, parasitic inhibition in vitro, and in vivo activity, and we did also some physicochemical characterization of the effect of liposomes to try to understand the mechanism of especially of the liposomes effects. So we have worked with Amazonensis, so that adds to that list a new species that is susceptible to topical miltefosine. What we have, I will tell you only what is really struck us as strange or notable. First, that if you see here in the left-hand graph, which is parasitic inhibition as a function of concentration of miltefosine in vitro, you can see that here in this box, you can see that both miltefosine and pure fluid liposomes have parasitic inhibition properties, which was really strange. So at the lowest miltefosine concentration, we saw that the fluid liposomes per se had a parasitic inhibition capability. Also at an intermediate concentration. So this was a kind of a surprise. Although there is one other paper by an Argentinian colleague showing something similar. Then we studied all these different liposomes and these different relationships between the amount of lipids and the amount of miltefosine, and we chose, as you can see here in this arrow, we chose this formulation. So the miltefosine incorporated into fluid liposomes with a high miltefosine phospholipid molar relation. So little amount of phospholipid, high amount of miltefosine, because it had the highest efficacy in vitro. So we then went to the mice, and we got really extraordinary results. I mean, the lesion size went down until it disappeared. You can see here in the picture in the middle. This is the control on the left, and on the middle, animals treated with miltefosine or with miltefosine in phospholipids. And here is the other thing that is really strange, or not strange, but it draws our attention, because this animal here in the middle has only been treated with miltefosine 0.5% alone. So there is no penetration enhancer, no liposomes, no DMSO, no nothing. It's just miltefosine on a gel, and it cures. It's amazing. What do we need the liposomes for then, if miltefosine alone can do it? Well, when you add the liposomes, you have the same result, only faster, right? So if you see here this graph of parasitic inhibition at the end of treatment, you can see that immediately after the end of treatment, if you take the skin of the animal and you reculture it in a way that the parasites can revive, if they are still alive, if they are still viable, you can see that immediately after treatment with miltefosine, you can still find viable parasite in the skin. But if you have treated the animal with miltefosine and liposomes, you find no viable parasites. So the liposomes kill the parasites faster. Also, we studied recidiva 30 days after the finishing of the treatment, and we found that there was no recidiva in any of the animals. You can see that after 30 days, animals treated only with miltefosine do not have any viable parasites anymore. So miltefosine cures by itself. You can just use miltefosine, but if you add, and you will eventually eliminate the parasites after 30 days. But if you use the liposomes, it will be faster. So then we did some physicochemical characterization in front cells. We saw that in the top graph, you can see that miltefosine alone goes through the membrane that imitates skin faster than miltefosine that is incorporating into liposomes. So liposomes slow down the miltefosine diffusion. This is confirmed by the smaller slope in the Higuchi model. But we also saw that our liposomes are not stable in size. So they start small, 100 nanometers, but then they fuse with each other quite quickly. But this is, well, we can discuss it later, but this is, I don't think this is too important. And we found no degradation after 90 days at 4 degrees Celsius, which is also a very important thing. So our hypothesis on the liposome mechanism, why is it that the animals cure faster when miltefosine is on liposomes? Well, we think there's two reasons for this. One is that the liposomes produce a depot in the skin from where the drug is slowly released. But also we think that the lipids themselves have an effect on their own, based on our in vitro results. So I would like to mention the challenges. So this is all very nice, but of course mice skin is much more easy to penetrate than human skin. It's at least 3 times more easy to penetrate than human skin, at least 5 times thinner. So we definitely need to prove this either in animals with thicker skin or in humans. The other issue, of course, is the stability, the tropical stability of this formulation, because the liposomes have unsaturated chains, so they would easily oxidize at high temperatures. Then, of course, the great advantage of this would be that if you can keep the drug in the skin, you would have no teratogenicity, for example. But this has to be evaluated, right? So the other thing is that this would only be useful for localized cutaneous leishmaniasis. So for metastatic species of leishmaniasis, this would have to be used in a different way. So I think also of course for co-infection with HIV or some other form of immune compromise, a topical treatment would have to be used together with a systemic treatment. And in this case, I think the topical treatment would be good because it would allow, I think that we have to prove this, but I'm optimistic that it would allow a lower dose of a systemic treatment, and then maybe we could reach all of the patients. So that's all. Thank you for for your attention, and these are my collaborators and the funding.
Okay, so thank you very, very much for that, Dolores. There's a fantastic overview of the topical treatments out there. Very interesting that you mentioned HIV there at the end as well, which I'm going to come to in the Q&A. In terms of the Q&A, I'm just going to stop your slide presentation now. If I could ask all the other panelists to come back. Thank you. There's Byron. Thank you. And I think there's Fabiana just joining us. And I know, Dolores, you are there as well. Feel free to put the video on, Dolores. We can see you when you speak, if you're happy with that.
Yes, I would if I could, but my connection is too bad.
Oh,
I'm sorry, you will have to do with my picture.
That's no problem. It's a great picture, so don't worry. It's brilliant. Well, the first question, we've got a lot of questions coming in. So I know this particular aspect of the NTD world is known fondly as the Leishmaniacs. So I was expecting some real technical Leishmaniac type of questions, which are definitely coming through, which we'll come to in a second. But from the ISNTD's perspective, and really from the perspective of the WHO NTD roadmap, shifts that are contained within the roadmap to 2030, and how they're going to impact research in this particular field. All of that, coupled with the fact that you both mentioned HIV, the immunocompromised situation, both at cutaneous leishmaniasis level, as well as the co-infected reality for the VL sufferers that Fabiana kind of alluded to. What I wanted to ask is, one of the major shifts in the roadmap is this kind of move from a vertical disease setting to a more holistic, cross-cutting, cross-disease approach. So as an NTD community, we need to reach out and speak to different diseases. You've all mentioned HIV. So the first question is, and I'm going to come to a platforms question after that from Maria Paula, but I think the first question to do is really from your perspective, Fabiana, from a VL HIV co-infected perspective, what would be your message to the HIV community?
So for HIV VL patients, definitely the integration of the care for the patient is of paramount importance. So the first thing that we always say, and it's already in the guidelines, that as I mentioned, the management of HIV VL co-infected patients has to be very different. So the first thing is, for every VL patient, it is very important that HIV test is done. So the first thing is to identify who are these patients who are the co-infected. And then the other way around, in the HIV clinics, they have also to think about VL as one of the possible opportunistic diseases, right? This is very important that it is included in their, organigram on how they assess the different patients, especially for those endemic areas. It is important that a patient who's being followed in an HIV clinic that starts to have symptoms that are suggestive of VL. Remember, there is some tricky things there because, for example, one of the most prominent aspects of a VL patient is the large spleen. But in HIV, not always they have large spleen. And there is also the complication that for HIV VL patients, the current diagnostic tests are not so good as for immunocompetent patients. So all of this has to be taken into consideration for us to, first of all, identify who are these patients and then be able to provide the proper treatment to them. And then I think the other thing, Cameron, as I said, it's not just to treat the immediate active VL case, but then it's also providing a very important, the proper follow-up with active ART, switching to second line whenever you see patient that, CD4 keeps going down, viral load very high. So this integrated approach for HIV VL, no doubt, it is the only way that we can, give a little bit more of survival for these patients.
Okay. So thank you. So ultimately change the treatment algorithm, the treatment pathway by integrating the HIV community into this. That's quite a strong message. I ask that because you get a lot of, another totally different disease state, FGS, female genital schisto, that also speaks to the HIV community. Those people with FGS go on to high risk of getting HIV. And when you look at the funding behind global health, most of the funding goes to the HIV or the big three, TB, HIV, malaria. I'm just wondering what kind of messaging can come out of these programs to bring that kind of community into play. So that's a very good point you've made about integrating the treatment pathway, changing the algorithm as the patient presents themselves. So thank you for that, Fabiano. May I ask the same question to both Byron and to Professor Dolores Carres, please, because you both mentioned HIV from an immunocompromised situation. What would be your message to that particular crowd?
Byron, you go first.
Yeah,
you'll have to unmute yourself. Sorry, Byron.
Sorry, the technology is winning here. I think there is not much difference with what Fabiana has already mentioned for the CL, even though I must say this co-infection HIV and CL is much less dramatic, let's say, than in BL. And of course, basically, when it comes dramatic is when you see this patient with this special form of cutaneous leishmaniasis, like disseminated cutaneous leishmaniasis or diffuse cutaneous leishmaniasis, where the immune response from the host somehow triggered by the same infections or associated with infection is also put in big fight with the immune response or the immunocompromised host with the HIV. But for localized cutaneous leishmaniasis, where the number of cases co-infected with HIV is relatively small, there is not much problem. Nevertheless, the approach that Fabiana mentioned, and that is what is in those cases are being followed, similarly also for mucocutaneous leishmaniasis, I should say, that all patients with this special form of leishmaniasis are more or less by routine also tested for HIV, just to see if there is any kind of co-infection. And the vice versa is also true. So if you see patients with HIV who has some suggestive lesions associated with leishmania, then they are also tested for leishmaniasis. That's what I'm trying to say.
And Dolores, do you feel the same? I mean, I think you're alluding there to a knowledge gap, potentially, Byron, in terms of these lesions, if they're picked up by the HIV treatment pathway, can they be related back to you guys? Perhaps you're alluding to a knowledge gap at the HCP level, but Dolores, what's your view on that in terms of the HIV crowd? What would be your message to them? How can we leverage that massive community that have got probably reams of data in terms of patient data, reams of engagement with these communities, all sorts of financial cushions in terms of the backing for HIV? How do you reach into that particular audience with your? It's
the question, right? So. Here, I have noticed that here in Argentina, I don't really know. I guess in Brazil it's different. In other places it's different. But here in Argentina, most people that I talk to about leishmaniasis, I'm working on this, and I'm so excited, and they don't have a clue what it is. They don't know what it is. They don't even know that leishmaniasis is an endemic disease in Argentina, which is not the case, for example, with Chagas. For some reason, all the communication, all the preventive medicine with Chagas has been much more efficient, at least when I was a child, right? So I learned about Chagas in primary school, even though in my city there is no Chagas. In my province there is no Chagas. It has been a huge problem in other parts of the country. So here in Argentina, Leishmaniasis is a huge problem in the north and in the northeast of the country. But here, south of that, and of course, we are richer, right? More in the center near Pampa, where we produce all the cows and all that. We are rich, and we don't even know about Leishmaniasis. But I think the important message here is don't be so happy about this, because there has been a couple of cases of cutaneous leishmaniasis indigenous to my province in the last 4 years. So global climate change, global warming, is bringing tropical diseases further south to the rich cities, to Buenos Aires, to Córdoba, to Rosario, to Mendoza. So I would use that so that people would really care, because people don't care. People don't know, because they think, oh, this will only happen to people in the north. We are indigenous people and we don't really care. Because that is really what is happening. I mean, they won't say it, of course, but that is what
I think that's a very honest, quite brutal revelation there, but I applaud you for saying that. What's very interesting is COVID has really shone a light on social inequality in communities, right? It's really shown that poverty is linked to diseases in a big way. And perhaps we need to redefine the social determinants of poverty to make these awareness campaigns trigger earlier. You mentioned climate change is one of the new kind of determinants, migration caused by drought, caused by climate change, that's entering into the equation when it comes to defining poverty, which I think will, in time, redefine the advocacy required around awareness for perhaps cutaneous leishmaniasis and other diseases of poverty. Thank you very much for those answers.
I would like to tell something more, that from the point of view of the discovery and development of new drugs that can combat, are active against cross infections. The concept is and this very, let's say, advanced concept to try to hit both infective agents with one, combined drug. And this is a problem of engineering in drug design and finding a protein and a target that can be relevant for both in some in some way. It's of course, in perspective, the use of this kind of drug can be of course difficult, but it's something that is in the way, on the way. So that just to say that.
Well, to be honest, Maria Paula, I was about to ask you that question in a different way, which was this. That's absolutely no problem. I'm glad you said that. I'm very glad you said that. What I was going to ask you is that obviously that shift in terms of the WHO shift from one disease vertical to horizontal cross-cutting, what it really means is new platforms, new platforms against an existing disease, which would elucidate mechanism, would open up the landscape in terms of new targets, perhaps yield a better portfolio, different mechanism of action in terms of trials that are going on. So what I wanted to ask you was, from a discovery perspective, what you mentioned repurposing slightly in your slides. You mentioned some of the post-HTS, high throughput screening kind of approaches that you're looking at. What platforms have you got your eyes on that would be game-changing to this horrible question, but I think it's one that needs to be asked. From a discovery perspective, what are the platforms that if you had a blank check, you would say, I need this, this, and this, that would really help? What would change that landscape?
Yes.
I'm talking about the paucity, the paucity of the portfolio of drugs from a mechanistic perspective. There are not very many of them. What would change that? What platforms would change that?
Sure. No, it's an excellent question, because in fact I was provoking this through my presentation somehow, because there is this let's say shift between what is needed in drug discovery nowadays for a very up-to-date and perspective and to give a possibility to have a better drug with respect to the past, and the real affordability of these technologies. So it's a big gap, but this gap is not connected only with parasitic disease or vector-borne disease or neglected tropical disease, but also with all the mostly of all the other diseases. So they share problem, because some technologies like the omics technologies and also this kind of artificial intelligence that require very huge and powerful computer to analyze all data from genomics, proteomics, all these are very costly and no one single institution can afford this. Also the high-throughput platform for the machinery for, so costly. They cannot afford. So the concept is that, from what I can see and in perspective, is that all the different labs, of course, apart from joining in networks, and as we are already doing, okay, maybe it's not enough yet, but we can do that, is to identify a plan in which we have first databases that means having new chemical entities and known chemical entities. And these databases are all over the world. They exist and need to be identified and then applied for screening. Those centers in general can help in this, and they give back one lead for optimization or something different. And so the labs can go on. And then omics, so you have assays first against one target, and the target need to be, identified, but let's say the existing targets, or new target can be, tested, and proteomics and also through different technologies can be identified in collaboration. And this open, the way to the study of the mechanism of action using specific probes, for example, derivatives, helping with the medicinal chemistry. And this center can do that. Also, for example, crystallographic center, structural biology center, also, in UK, but everywhere, many places. So it's a huge, so summarizing databases, omic technologies, and high-throughput screening, this artificial intelligence, these are, let's say, pillars now, and they are available. Somehow they are available. Some work, some application, young people, young PhD and young students can apply from everywhere. So this is something I foresee in the future and the near immediate future. And some of them are looking for projects. So that's a lot of effort from the side of researcher, but that can have a successful output, and we need to do that, and we need to be more also, because there is no way up to when we have only not enough people working in the field. There is not enough critical mass, and this need to be done.
Well, I was going to say thank you very much for that answer. We have got a lot of questions in the chat box which I can't actually access right now for some reason. I think that's what happened with the system when it configured. I think that was a great answer, actually, Maria Paola, because we are really trying to assess the potential impact of these the shifts in the WHO roadmap, especially to this area of Leishmaniasis. One from Dr. Obaid Hayat from Pakistan, from where Leishmaniasis parasites come to the fly guts? That's one question, which I'll open to the floor.
They are taken from these female sandflies when they are feeding from the animal reservoirs.
Okay, thank you.
Those are the vectors who transmit then the disease to humans.
Or from patients.
Okay, so Dr. Paul Cole from the University of Kent is asking, are other domestic animals or livestock also potential reservoirs of infection? And does the specific local sandfly vector population play a role in whether transmission occurs or not? Yeah. So are other domestic animals or livestock also potential disease reservoirs?
Yeah. From what I know, yes, sure. Okay, it's written also. Dolores already answered. Yes.
Yeah.
It's cats, for sure, rodents, rats, horses, sheep.
Yeah.
Yes.
So, Dolores, would you like to answer that, perhaps, in terms of a wider answer for the group, so we can all hear it?
So, Byron,
no?
Can you?
No, you have already mentioned that there are several mammals that are being identified as reservoirs. And as Fabiana mentioned, for the case of PKDL, the humans can also act as reservoirs as well.
Yes.
Yeah. But I think if we talk about domestic animals, I think especially for VL, dogs are really the main ones.
Yes.
Because I think the question was more about domestic.
Okay.
So the transmission, the direct transmission is not something that occurs easily, but some cases. But of course, through the fly, it's possible, vectorize the parasite as also asymptomatic patient. Also, these patients are somehow among the reservoir, let's say. Yes.
I think if I may comment on that one, there has been a lot of work towards this aspect of the different reservoirs of the disease, especially for Eldonovanni and tropanotic disease. There was, the mathematical modelings were bringing always the question about asymptomatic individuals. So there is a recent publication from the group of Professor Shyam Sundar, O.P. Singh, which is, I would recommend for those who are interested to look at. So they have done diagnostics for the three main patient populations. So active VL cases, PKDL, and asymptomatic carriers that they followed up in the communities. And the main conclusion of the study is that the most important reservoir are really the active cases. So that's why you have to identify, diagnose, and treat as soon as possible. PKDL patients are very important reservoirs, and they were not able to actually have a sandfly infected when they used the asymptomatic carriers. So really this new study, I think it's quite important because it is showing that that hypothesis that asymptomatic were important now is under question. I think mainly the focus should be, active cases and PKDL, especially I'm talking about anthroponotic, right? If you talk about zoonotic, then there is the whole world of the animals, which makes the whole completely.
So thank you for clarifying.
Thank you for that. Thank you very much, guys, for a very comprehensive answer to that question. We've got an answer from Mochi Jignesh Kumar from the University of Gujarat in India. Asking, is there one metabolic pathway protein that's chosen as the effective drug target against VL? Is it enough to reduce VL infection? Is there one metabolic pathway protein that's chosen as an effective drug target for VL? Will it be enough to reduce VL infection? So I suppose that's to Fabiana and to Maria Paula.
Please, Fabiana.
Yeah, so I think for Maria Paula can give more information about the early phases of development, but I think the most important thing that I always hear from our colleagues from Discovery is that there is a need to identify new mechanisms of action. So by having different mechanisms of action, you can, act on the parasite in different pathways, especially if we are thinking that for some regions like Africa, we have a very high bar. We need to have treatments that are 95% efficacious, and that can be treated in less than 2 weeks. So that's where it comes the concept of possibly having a combination. So when you have two drugs with different mechanisms of action that are acting in different pathways of parasite, there are the advantages of, probably being more efficacious. And one thing we always say is that you have to hit hard and, reduce parasite load as soon as possible to break the cycle of transmission. And mainly, the idea is that by reducing the parasite load as soon as possible is how you would have an impact on the transmission side.
Yes.
Don't know, Maria Paula.
And that makes a lot of sense, Doctor.
Yes.
Yeah.
So I agree with Fabiana, because of course there are a few metabolic pathways that are, let's say, to work with, and drugs against those pathways are, important, because Leishmaniasis depends on the type of Leishmaniasis, but in general, they are very sensitive to that. But the most relevant point is to combine drugs, because this is basically is an infection, and it is already at this time we know that to combat an infection we need a multiple drug combination, because it's not you need to block more pathways, not just one, in order to reach the complete killing, and also to avoid drug resistance. And combination is not just association, that is different. So to find the right combination, this means to do some studies about that, and to find out the drug that we don't have enough drug to do this in this way at the moment. But in the future, the concept is to find a combination that act at lower concentration, lower doses, with in a synergic way, So you give less, but they are synergic and give the same effect. So you decrease resistance development, and you have compounds that are less toxic. So the principle.
And I saw that in your presentations as well, from all of you, about this redefining repurposing, actually, by using combination trials, repurposing of the drugs in combination with existing drugs for the trials. Very interesting points raised and echoed somewhat by the attendees. So Dr. Obaid Hayat has said, is it possible that one compound or drug target is it possible that one compound or drug targets two or multiple pathways at one time? I think you've answered that to some extent. Christopher Newton is asking, what is the MOA of the pyridine compound identified from screening 500 compounds against 6 parasites? Is it active on all parasites? So there's quite a specific question there.
This compound is active against Trypanosoma brucei and also Leishmaniasis. Yes, against both, and but we don't know the mechanism yet, because this is just a new compound. So we identified it through high-throughput screening. So it's not developed yet the convolved the target. So we are studying that.
Dr. Eric Schwegler from the University of Maine has put a statement saying an interesting metabolic pathway is the unique peroxidized clearance cascade in kinetoplastids. Drug targets in this metabolic pathway are triparadoxin and trypanothione reductase. So perhaps potentially someone to liaise with and potentially find some collaborative space. I mean, we've got to get through, but told that the session is going to auto end shortly. We're 15 minutes over the session. There are so many more questions to ask, but I think we're going to have to call it a day at that. It's been a long day in terms of D3. We're delighted to have this particular session at the end of forward movement in an area of quite high neglect in terms of Leishmaniasis. And it does encompass all the other sessions that we kind of covered in the day. People are coming back with thank you everyone for an amazing session, an amazing day, fantastic presentations. So you're getting a lot of claps actually from people, and that's echoed from us here as well. I wish we had longer time to discuss, especially some of these issues around mental health, stigma, already the high toxicity of these existing treatments. Is that going to make people not come forward when you get into the rural communities and you're trying to show them a new drug? Are they already kind of conditioned, thinking it's going to be highly toxic? So loads of stuff to kind of talk about on that. But I think from us today, I'd like to say thank you very, very much. Eric Schwigler saying, I learned so much today. Thank you for organizing and moderating. Annabelle Zabala is giving a clap. Helen Price from the University of Kiel. Thank you, everyone. Wonderful session. Paul Howe from the University of Kent. Thanks, everyone, and what an amazing day. So thank you to Fabiana. Thank you to Maria Paula. Thank you to Byron, and thank you to Dolores. Okay,
Thank
so
you, Hannah. Thank you.
Thank you.
Thank you so much for the organizing.
Bye-bye.